Thanks, Edmund. Thank you very much for the very kind introduction. As Edmund said, today I'm going to talk about one aspect of my research, which is pretty much drug delivery of challenging therapeutic molecules. I also have several other projects more in the pharmaceutical manufacturing of small molecules and biologics in terms of kind of material properties and process analytics. And Stan, who's talking after me, will talk a little bit about the project he's just starting now in our team. So most of you are probably very aware, given how fast the COVID vaccine came to market, of the long timeline for when you develop a molecule to a medicine from the point of drug discovery through preclinical trials, clinical trials, and until you get FDA approval. So for therapeutic molecules, which are a little different than vaccines, on average, it takes about 15 years to get an FDA-approved bioactive form from the point of drug discovery. So what I kind of hope our research contributes to this challenge, and I consider it a grand challenge, this length of time and the cost and expense of bringing a molecule all the way through this process. I hope that we'll be able to potentially shorten that timeline, that our work will contribute to that, and our focus is really the use of extensive pre-formulation strategies. So that's what I'm going to talk about today. Basically, what I want to try to do with my research is to try and reduce the attrition rate of molecules that are discovered with a relevant therapeutic effect, reduce the rate at which they're discarded during this whole development process. A lot of drugs fail when they reach clinical trials. Sometimes they go back and they get reformulated, or they're just abandoned altogether. And that's takes a lot of time, costs a lot of money, as I said. So our approach is to characterize the physical chemical properties of these challenging molecules. And as time goes by, it seems the molecules that are of most interest or have the most interesting therapeutic effect, or that are discovered from high throughput screening processes, do have challenging physical chemical properties. And then we want to try and manipulate those properties by designing either the solid state or the formulation and solution of these molecules. And then we want to do as much testing as we can, both in vitro and first of all, and then of course you have to test in vivo to see what's happening. The types of molecules we work with range from small molecule. Typically, I focus on small molecules with either poor solubility and or poor permeability. So two examples here, you can see clofazimine on the left and paclitaxel. You'll see throughout my talk that quite a few of the molecules I work with are antimicrobial molecules. So here, niacin A on the top right, and thyrosin and lactocin, they're all different types of bacteriocins. So they're peptides that are produced by bacteria to kill other bacteria. And these bacteriocins, we collaborate with a group in Cork, and they've been working on these for many years, but we're interested in their solution, solid state properties, and as I said, pre-formulating them. Two here you see thyrosin and lactocin. They're actually, so say if you take lactocin consists of two different peptide chains. They're called lactocin alpha and lactocin beta, and they work synergistically to attack the bacteria. We also work with even larger proteins, so things like C5a peptidase here, the enzyme on the left, has a molecular weight of about 104 kilodaltons. So this is a protein that Jackie Cooney and her team have worked with for many years, and they've elucidated the structure and the impact of changing a few residues here and there. Why we're interested in C5a peptidase is that it cleaves C5a, human C5a, and actually we've some interesting data that it cleaves a couple of other proteins that are part of the complement, your complement activation. So it's basically your body's response to infection or the things that cause some of us to have immune disorders like chronic diseases like psoriasis or Crohn's disease. So this enzyme has the potential to have an impact on the progression of those types of events in your body. I'm not going to talk actually much about C5a peptidase today, mostly because despite being a big molecule, it's actually quite stable. It's quite stable in PBS. What we are developing are controlled release systems like hydrogels, and Pratik, one of my postdocs, is working on this to control the release of it and get a prolonged activity over time. And just to note, we produce this in-house, and Vinay and Francisco, again, another pair of researchers in my team, they have been producing this in E. coli, again, from carrying on some of Jackie's work, and they can produce about 100 mgs in 2 weeks, which those of you who work with small molecules realize 100 mgs is not a lot to do a lot of experiments with. It takes a lot of effort. The lactocin on the previous slide, actually, we produce maybe in a week and a half about 5 mgs of this. So just to make you appreciate that you have to be very careful with your design of experiments when you have limited material available. On the right here is GAPTH, another enzyme that we work with as well in the lab. So lots of different molecules with different properties. So we study their solubility, we study their solution behavior, be that in plain water, but also in formulation media and in simulated body fluids. And then we study their solid state behavior if we're, and this is important, not only if you're administering the drug in a solid form to the body, but also if at any stage of your manufacture or production process, you need to isolate the drug molecule to dryness. And then we keep an eye consistently on activity. So if I start with one of the smaller molecules, clofazimine, so here's its chemical structure. It's a very insoluble drug. This is the neutral molecule. There's a couple of polymorphs of this that have been reported, but it is an ionizable drug, and it's a weak base, so you can protonate it and form salts, which seems like probably the simplest way to try and improve solubility. It's class 2, so it's a BCS class 2 drug, which means it has low solubility and high permeability. And it's marketed actually in a physical mixture with another drug in a product called lamprene, which is used to treat leprosy. So one of the PhD students, Porrick Bannigan, he's now in Toronto, but he worked on clofazimine during his PhD. And you can see here one of the first things he did, he made a series of novel salts and he checked their dissolution and their solubility behavior in water. So you can see here the citrate salt over on the right, you see the red solution, it dissolved fastest. Then the hydrochloride salt dissolved in water, but our polymorph, our neutral molecule, the form 1 over on the left, you can see none of it really goes into water. So you might think from this that, yeah, salts, you're going to get high solubility in water, which you do. Here's another salt. This is the phosphate. Goes into solution. I'll just get my pointer, sorry. Goes into solution. You get quite a high concentration of clofazimine, up to about 160 mg per liter. But when you look at the solution concentration of clofazimine from the phosphate salt in gastrointestinal fluids, and lamprene, the marketed drug, is taken orally, so we're looking at oral administration of the drug, you can see that the solubility decreases significantly, both in gastric fluid and intestinal fluid. And remember, your drug has to be dissolved for it to have a therapeutic activity locally and to pass through the intestinal walls and get systemically distributed in the body, so to have a good bioavailability. In fact, if you look at clofazimine citrate, again another salt, and you did a study on its solubility profile with respect to pH, you can see here that it has the highest solubility at a pH of about 3 to 4. But when you consider that the pH of gastric fluid is about 1.6 in the fasted state, and the pH of intestinal fluid is about 6.5, it makes you wonder, well, is this drug going to be absorbed at all in the body? Because it's not soluble at the pH in gastric fluid and the pH of intestinal fluid. We studied the solution behavior, the dissolution in the gastric and the intestinal fluid, and we also did it in this dynamic dissolution setup. So we added our clofazimine with water to our gastric fluid, then we added that gastric fluid after 15 minutes to our intestinal fluid, and then we added and we monitored the concentration of clofazimine over time. Now, Given the pH of GF and IF, you might think, well, we won't get anything in solution. What we found was, and this is a bit of a busy graph, but basically this dotted line is the point at which we mix the GF, the gastric fluid, into the intestinal fluid. And you can see you actually get quite a high solubility, a lot higher than we would have expected given the pH in the gastric fluid. Remember, it's 1.6. So we get a lot of clofazimine into solution. And the reason for this is that despite the low pH of the gastric fluid, the presence of pepsin, which is you produce in your body, actually solubilized the clofazimine molecule to an extent. And what we found was when we did the calculations that pepsin was actually binding 11 molecules of clofazimine for every 1 molecule of the pepsin. When you mixed it, you then with the intestinal fluid, you obviously diluted your sample, but you can still see you get a concentration of about 20 mg per mil if you add an excess of pepsin. Here we've added 125 mg of pepsin to the whole dynamic dissolution setup. So the pepsin actually improves the solution concentration or increases the solution concentration of this poorly water-soluble drug. So in fact, you might think that the neutral clofazimine molecule, that's never going to dissolve in water from the initial video I showed you. But actually, when you consider the composition of the environment that it's going to be in the body, there are a lot of components that could improve the solubility of your molecule, your drug. Similarly, if you look at lactocin, this is this bacteriocin, this dual-acting antimicrobial peptide, the 2 peptide strands. And on the left here, this is the lactocin alpha peptide. And you can see the large standard deviations. We get a little bit of lactocin alpha dissolving in our gastric fluid and our intestinal fluid, but it's extremely unstable. Okay, so it basically precipitates out very quickly. So the alpha chain is actually quite hydrophobic or quite insoluble in water. If you look at the beta chain, you can see very little solubility in PBS, that's your phosphate buffered saline, very little solubility at low pH again, but you get a much higher amount of the beta chain into solution in the gastric fluid and in the intestinal fluid. And we believe this is due to the presence of the phospholipids and the sodium taurochlorate in these media, which is in your body as well, in your gut, and it solubilizes the beta to a certain extent. Now, keeping in mind that the alpha and these beta chains work together to kill bacteria, we need both to be in solution to have activity. The other problem with lactocin is this is just a simple graph where on the y-axis you have OD, so that's your optical density. And basically what we do is we add a small number of bacterial cells so that they have an optical density of less than 0.1 to start with. And then over time, the bacteria start to multiply and you see they start to make your solution opaque, so your optical density here, you can see it increases, and this means that your bacteria is growing. Okay, so the dashed gray line is just your control and your bacteria is growing. And if you look in the presence of the lactocin alpha and beta chains, we get no bacterial growth. That's the gray line at the bottom. And in the presence of the lactocin and pepsin, we still see no growth, which means the lactocin is still active. But in the presence of alpha-chymotrypsin and trypsin, which are proteases, and they basically chew up our lactocin, and the bacteria grows fine. So our lactocin is susceptible to degradation by enzymes, which means we need to protect it when we administer it to the body. So they're just 2 examples of molecules where we look at their solution behavior. And then we start to try to manipulate this behavior to optimize their activity, their stability, and their potential to have a therapeutic effect when they're administered to the body. We do this in a range of different ways in my group. So we have a host of projects that look at tailoring crystal size distributions to product performance. We use supersaturating delivery systems. We've just started to do a bit more work on multi-drug therapies, and we want to design formulations or solid or solution forms that improve on the properties of physical mixtures of the drugs, which tends to be the way they're often administered. And then we control this behavior by integration of the therapeutic molecule into hydrogels, polymers, lipids, and ceramic matrices. Okay, so that's either by encapsulation, adsorption, or coating. So I'm not going to talk about all these projects today. This comes under the umbrella of the SSPC, but this is where we focus on making different polymorphs or different solid forms of the drug molecule itself. We focus a lot on the crystallization process. So we look at heterogeneous crystallization, we look at solution clustering and control over the nucleation process. Again, always trying to tailor the particle size of the drugs and the polymorphic form to get the product performance that we want. And when I say product performance, that means during the manufacturing as well as after administration to the body. We also do a lot of work with the anti-solvent precipitation technique, and hopefully over the course of the next year, you'll see some of these presentations from the various students from the Long Act Now Marie Curie project and from the SSPC meetings. So we do a lot with hydrogels. Some of you may have seen James Flynn's PhD presentation back in October, but today I wanted to focus on the work we do with lipid-based drug delivery systems. So lipids, and I suppose I picked this as well because you're all familiar that a solid lipid nanoparticle is used in your COVID vaccine. But lipids are non-toxic. They can improve oral bioavailability. They help with permeability. And they can be cheap. Now, not all lipids are cheap, as you'll see in a few minutes when I start using monoline and phentantriol, but there are some very cheap lipids available. And they can be formulated into a range of different types of particles for drug delivery. So Avian, who's in her 4th year of her PhD in the Bernal and in the Chemical Sciences Department, she encapsulated her lactacin into solid lipid nanoparticles. Now remember, lactacin consists of your 2 peptide chains. So she took 2 different approaches. So one, she put 1 peptide chain into a separate solid lipid nanoparticles. And in another strategy, she encapsulated both the alpha and beta chains of the lactacin into 1 solid lipid nanoparticle. Finding the composition of the solid lipid nanoparticles is not always obvious, and Pratik, a research fellow in my group, would have helped Avian a little bit with this, but she used a lipid system, a lipid softasan, and then she screened a range of different surfactants and co-surfactants, so colifor, transbutyl P, polyethylene glycol, polypropylene glycol, with different molecular weights and at different weight ratios to the lipid. And then she screened a range of different solvents because we needed to dissolve the peptides to encapsulate them into the lipid nanoparticles. So you can see her optimal formulation is colored in gray here. So it's the colifor RH40 in a 1:1 ratio with 100 mg of lipid with the transbutyl P and using a small amount of acetone and DMSO in the process. She got both in encapsulating the peptides, she got over 90% encapsulation efficiency, and the loadings of the peptides were roughly about 9 to 18 mg of peptide per 100 mg of lipid. But what she also managed to do, which was important in the doubly loaded solid lipid nanoparticles, so the particle where you had both peptides in the same particle, she had an equimolar amount. So she had the same number of peptides of the alpha peptide and the same number of the beta peptide in the one solid lipid nanoparticle. And here, so again, this graph, for those of you who are not familiar with growing bacteria, on the y-axis, you have the number of colony forming units, so the number of colonies in a log scale. You have the bacterial control, so this is how the bacteria grows and the number of colonies it would grow during the experiment. You can see the blank SLNs don't have any impact on bacterial growth and they do not kill any bacteria. And then you can see we have different concentrations of lactacin. So it's written in decreasing concentrations. So the highest is here on the left. And you can see the gray bars here are your free alpha beta peptides. So they're just added to the media without any lipid matrix. And you can see these large standard deviations, and we think this is also due to the inherent instability of these peptides in solution. But they're definitely a lot less active against the bacteria at the lower concentrations than the peptides that are encapsulated in the lipids. And interestingly, you get a better killing from the peptides that are encapsulated into the same solid lipid nanoparticle at a lower concentration than you do with when they're incorporated individually into each solid lipid nanoparticle. We looked at the release. So again, here your number of colonies in log form on your y-axis, your control, and here we were looking at the release of the peptides through a membrane. And you just take the supernatant and see if there's activity in the supernatant. So the lactocin is being released from these solid lipid nanoparticles, and this was done in IF, so it was released into intestinal fluid, which contains some lipase, which do degrade the solid lipid nanoparticles, and you have activity for about up to 48 hours. After 48 hours, we start to lose activity. And remember, this is a log form, so this activity is actually, it's killing a lot of bacteria. So then another type of lipid system that we look at are liquid crystalline phases or lipid cubic phases. And basically what we do is we take a particular type of lipid and we melt them and we add water and we make sure we're in this part of our phase diagram, so kind of above 30% weight per weight of water, and we form cubic phases, be it the PN3M or the IA3D cubic phase. And you can see here, this consists basically of a membrane formed by your lipids and then water channels. And the beauty of this is these liquid crystalline phases is that your hydrophilic molecules will be encapsulated into the water channels and your hydrophobic molecules will be encapsulated into that lipid membrane. There's a range of commonly used lipids. These are a little more expensive than, say, the softosan that was used in the solid lipid nanoparticles. The work I'm going to talk very quickly about is used mostly monoline. Like I said, here you can see some of the hydrophobic drugs that we added to the molten lipid, and then you add water to form your hydrogel. If our drug is hydrophilic, we would add the drug to the water phase, and again, then add that to the molten lipid, and you form this gel, which has a consistency a bit like toothpaste. We can break that up and homogenize it if our drug is not super sensitive and form cubosomes, and you can see the solid dispersions down here. We also use this system to encapsulate enzymes, and we saw that the enzymes which basically lost activity usually after 3 days in solution were stable upon release from the gel for up to 10 days. But we don't do homogenization with our enzymes because they would not like that, and we used different solvent evaporation technique to form cubosomes in that case. We characterize these cubic phases by small angle X-ray scattering, cryo-TEM. Now these, we have to go off campus for these obviously, but we do check that we have a crystalline gel by using our cross-polarized light microscope here in the lab. And then for the cubosomes, we use the Z-decizer to measure their size and their zeta potential. I'm conscious of time, so I'm going to quickly over this. So Michelle did some very nice work. She just graduated last year. She's now working up in Pfizer. So she incorporated a lipase inhibitor. So lipase is an enzyme that's found in your body that will degrade lipids. Okay, so the lipase, you can see it here. This is this serine is part of the active site of the lipase, and it attacks the ester bond on monoline, and you get faster degradation of your gel, your lipid cubic phase, and therefore you get faster release of your drug, particularly the hydrophobic drugs that are encapsulated in the membrane. What she did was she incorporated a lipase inhibitor, tetrahydrolipstatin, and basically there was then competition for the binding site, the active site of the lipase. It bound either to the THL or to the monoline, and that essentially slowed down the rate of degradation, and so you could control the release of your drug. She did this by with clofazimine, the drug I talked about earlier. So you can see here the red lines have no lipase inhibitor, and you can see the dashed line is the rate of degradation of her bulk lipid cubic phase hydrogel, and the red line is the rate of release of the clofazimine. And so you can see that the rate of the release of the drug, because it resides in the lipid membrane part of these cubic phases, follows the degradation profile. With THL, the inhibitor, you can see she completely slowed down the rate of drug release and slowed down the rate of degradation of the drug, or sorry, of the lipid. She found that if she varied the concentration of her THL, she could tune essentially the rate of degradation of her gel, which essentially means she could tune the rate of release of any drug that's any hydrophobic drug that resides in the lipid part of the lipid cubic phase. But of course, a lipase, when it's in your body, doesn't act alone. There's lots of other components, and it boils back to this thing that what we do in the lab, there's so much more of a complex environment in your body, which may change the dynamics and the kinetics of transformations of your drug, of release of your drug from a formulation, etc. So Cheyenne, who works with is a research fellow with Damien Thompson, did some very nice molecular dynamic simulations, and he took into account that in the body you have your lipase. So the top row here is the lipase inhibitor interacting with the lipase. The bottom row here is the monoline lipid interacting with the lipase. But then he also factored in that you'll have another protein, colipase, which interacts with the flap. Lipases have a flap on them, and when the flap is opened, when it's near a hydrophobic environment, then the active site is available for binding. He also took into effect that another cofactor, calcium 2+, influences the activity of lipases. And he could model, and again, I'm not going to go into this too much, I don't have time, but he could model the binding energy of both the active site with the lipase inhibitor and the lipid itself, and the overall binding energy, the effect of those on the overall energy of the complex. And so it helped us realize what the competition was between the inhibitor and the lipid for the lipase active site. And then we did some experimental work in the presence of the cofactor, in the presence of lipase, the cofactor, the colipase, and calcium, and saw that it did change the rate of degradation of our lipid cubic phases. So very quickly, I'm going to go through stage 3 of the approach we take, which is our in vitro and in vivo testing. We look at activity in different cell models, cytotoxicity, pharmacokinetics in vivo, permeability, and in collaboration with Kieran McGorty, he is providing some really interesting in vitro models that we can test our drugs, our multi-drug approaches, and our formulations with. And we're working together on a skin model, a disease skin model to treat psoriasis, and also we're hoping to develop an inflammatory model in his intestinal organoids. But hopefully he'll give a seminar at some point in the future, and he'll talk a bit more about those. So we do what many people do, a lot of MTT assays, so studies cell toxicity. So here, this is just showing that the SLNs with the lactocin don't have any toxicity, or the cell viability of endothelial cells is fine over 24, I think this was 24 hours, no, 4-hour incubation. Here we looked at, this was Michelle Dully's work, where she took her cubosomes, those lipid cubic phases. They had a range of antihistamine drugs encapsulated in them. There's 4 different antihistamine drugs. One of the drugs, cetirizine, is the yellow one here. That showed a little toxicity against the fibroblasts, the 3T3 cells. And then she also tested the cell viability against a ORBL2H3 cell line. And she wanted to do this because it's a commonly used histamine releasing mast cell line. And so she tested the amount of histamine released from these cells, and she found that the drugs encapsulated in the cubosomes could reduce the amount of histamine released, which was a promising result for these systems. Another study here. This was where we used drug delivery stents. So when a stent is deployed in your body, this is in collaboration with Cook Medical, it causes injury to the inner lining of the blood vessels, which are comprised of endothelial cells, and you get inflammation and potential thrombus formation in the area. So PTX, paclitaxel, is a drug that's often loaded onto stents, and that stops cell proliferation, but unfortunately it stops both the endothelial cells from proliferating and repairing itself, and it stops smooth muscle cell proliferation, which is desirable. So Mariama did some nice work where she saw that citric acid, first of all, could reduce inflammation by lowering IL-6 expression, so a marker for inflammation. So you can see it there, but only at a particular concentration. But she also showed, and I'll just say it, I'm not sure you'll be able to deduce it from this graph, but essentially a mixture of the citric acid and the PTX could preferentially restore the endothelial layer and still suppress the smooth muscle cell proliferation, which is desirable. And very lastly, and I'm going over time, Edmund, I'm sorry, but very lastly, this is a study I did 14 years ago. I had been working with mesophase silicates for biocatalysis during my PhD, so nothing to do with drug delivery. But when I was studying these materials, I kept coming across lots of articles saying that they are really nice materials for using for drug delivery. The reason being they're powders, they have these ordered channels, and you can fill these pores or these ordered channels with drug and then control the release of them. Here, we still work with these, by the way, even though you'll see in a minute, they're not always the best for drug delivery. But you can see this is a nice TEM image taken by Jennifer Cookman on an SPA 15 material made by Marta, who is working in the group now. If we did cell toxicity assays on these with a range, these are just 4 different types of mesophase silicates, essentially all have this siloxane composition. The cells are doing fine. Another cell line, these muscle cells, C12s, see a little bit of a loss in cell survival, but nothing too bad. But when we put them into animals, if we put them subcutaneously, they were fine. If we put them intravenously or intraperitoneally, that's in the space around your intestines in your body, we saw benign local biocompatibility, but lots of the mice died. So we saw considerable systemic toxicity. From the histology that we studied, we suspected maybe thrombosis or complement activation, but we didn't know. The role of these mechanisms wasn't clear, and it certainly wasn't predictable from our simple MTT assays, cell viability assays. And I suppose what the point I want to make here, and this is my last slide, is that even 15 years ago, we didn't have access to the kits and the assays that allow us to detect the different cytokines that are upregulated or downregulated. And the ability we have now to do this helps us a lot in trying to determine if our pre-formulation strategy is going to be a valid one in the body. So that's basically it. Our overall goal is to develop rational approaches to reduce future attrition rates of therapeutics when you reach the clinical stage of development. Of course, apart from the last bit, didn't do any of the work really. It's all of these guys and my former PhD students. And I'd like to thank you and take any questions. And sorry, Stan, if I went over into your time.
Thanks, Sarah. That's a tour de force. That's a huge array of areas, and I know you already talked about a few of them. There's time for a few questions, so if anybody has any questions, I'm just trying to see if the chat. While I'm doing that, I have a couple of questions I wanted to ask myself while I'm seeing if I can see any hands up. How close are you and how close is the field to having a set of design rules where if you let's say Eli Lilly came in and they're making a brand new protein when they developed their plant in Limerick and they wanted to stabilize it. Do you have to start off from scratch with each one, or is there a kind of a set of that you can actually go down? Like there was a kind of decision tree. Like it was in one of the slides you had there. One of your students had highlighted a kind of a green color, the pathway that she took to stabilize.
Yeah.
can you go down that decision tree, or do you have to kind of do a screen and just see how it goes?
A lot of it, certainly in industry now, and I mean, proteins are obviously harder, but even for something as simple as forming a salt, it's still a decision tree. It's still a trial and error process, and that's what we would love to move away from. And that's, you know, it's funny. I sometimes wonder, you know, you see a lot of conferences with drug delivery titles, and you see a lot of conferences with pharmaceutical formulation, and when you talk to people who are in pharmaceutical formulation, they focus a lot on, you know, the molecular behavior, and in drug delivery they focus a lot on the interface between the material and biology. And I feel like there's the two need to talk more to each other. But in terms of, you know, a lot of companies have particular buffers that they know stabilize their proteins, and they will tend to go for what they traditionally have found worked before for them, and that's their starting point.
Yeah. How does, you know, the CDZ, I can't remember the name of the drug, the one in the first slides.
Yeah,
that obviously that's commercially available, so that's obviously stabilized in some way, so that it can be it can be absorbed in a smaller intestine. Is that proprietary, or do they reveal how they stabilize it?
Well, I think that the point is that it actually does dissolve. I suppose the point I wanted to get across was, if you look at its water solubility just in water, it's got extremely poor water solubility. The salt is better, but it's actually the free drug that's in that lamprey product. But the point is that in a biological environment, it actually does get solubilized, not through anything they have done. So failure, like a drug that may appear to be very poorly water soluble in water, there may be components in the biological environment that actually solubilize it, and so you get more across. But just to make a point on that clofazimine, it's used to treat leprosy. It actually turns your skin pink after a while because it accumulates. It's so hydrophobic, it accumulates in the tissue of your body. So it's not. You don't really want to take it unless you have to.
Yeah. Okay. Thanks so much. I don't see any hands up, so unless I'm missing them, any other question? Any from the floor? The one comment I'd say, Sarah, though, is that because it's unknown what you can for you know, it's kind of a trial and error. There's enormous scope then for work to be done.
Absolutely. Yeah.
And
I think that becomes more important as more and more complex molecules come to the market, like, you know, and especially for things like fusion proteins, and there's a lot of stuff that still needs to be done.
Yeah. Okay, thanks very much. And can I briefly say thanks for being the volunteer first?
I believe you volunteered me, Edmund. But
well, I did ask.
No problem at all. Thanks, everyone.
Thanks. Okay, we go to our second presentation, and this is from Dr. Stanislas Helle, who is a postdoc with Sarah. And Stanislas received his PhD from the University of Lille, and as part of his work there, he worked with a company, Florimond Desprez, and the focus of his work there was to identify biomarkers for sweetening potatoes at cold temperatures at less than 5 degrees Celsius. And from what the details he's given us, he's now he's obviously was a research engineer with the same company, but he's now bringing his expertise in proteomics analysis, and he's applying that to work here in SSPC in the Bernal Institute. So I'd like to, Stanislas, I'd just like to welcome you, and the floor is yours.
Okay, thank you for the presentation. Is everyone able to see my slides?
Yes, yeah, we can, yeah, thank you, yeah.
Okay, so good afternoon, everyone. I am Stanislas Helle, a postdoctoral researcher in Sarah's group here in Bernal Institute. The purpose of my talk will be the proteomics analysis by mass spectrometry. I will illustrate this presentation with some proteomics experiences I previously made during my PhD on plant biology, and some I plan to do here for biopharmaceutical research. So as a really short introduction, proteomics is the characterization of all the protein constituting either a cell or an organ or an organism, even a sample from a purification process. So proteomics is part of the large family of the omics, with the genomics, which is taking care of the DNA sequencing, transcriptomics, which is about the RNA produced from the DNA transcription, and metabolomics, which is about characterizing metabolites inside samples. So in order to analyze the proteome, for sample preparation, there is two main strategies. The protein can either be separated inside a gel, so it can be a 1D gel like SDS page, or a 2D gel with a separation by molecular weight or isoelectric point. Then the spots in the gel can be cut, and then each spot is analyzed with the protein inside. In this case, for the mass spectrometry, we will prefer MALDI-TOF as a mass spectrometer for the protein identification. Otherwise, the protein extract can be characterized directly without separation prior analysis. We talk about shotgun proteomics, and in this case, the separation process takes place during the analysis with the use of liquid chromatography coupled to the mass spectrometry. For the analysis itself, there is also two main strategies, top-down proteomics and bottom-up proteomics. So top-down because we go from the entire protein to the amino acid sequence, and bottom-up proteomics because we go from the peptides to the protein. There is also middle-down and middle-up strategies, which consist in the analysis of large fragments of proteins. This technique is useful for the analysis of large proteins such as immunoglobulins. Let's begin by explaining bottom-up strategy, which is the most used strategy for proteomics. So proteins are first denaturated, reduced, and alkylated in order to linearize it. Then they are digested using a protease. Most of the time it's trypsin. And all these peptides are analyzed by mass spectrometry. So the peptides, so for each peptide we will obtain a peak, which will appear a peak massif. So each peptide has his own peak massif. Thereafter, this peptide can be isolated and then fragmented and analyzed the second time by mass spectrometry in order to obtain the sequence of the peptide. This sequence is then used in order to retrieve the protein by matching the peptide, the peptide sequence with the entire protein sequence using databases. With top-down strategy, the entire protein is analyzed. This process is mostly done using MALDI-TOF. First, the protein is analyzed entirely in order to obtain the mass of the complete protein. Thereafter, the protein is fragmented in order to obtain its partial sequence in N-terminus and C-terminus by obtaining fragments. And this partial sequence, coupled with the mass of the entire protein, will help to retrieve the protein identity using databases. So during my PhD, I used proteomics in order to identify all the proteins sequestrated inside the potato starch granule. So this project was a collaboration with the MSAP, which is the proteomics platform of Lille, the glycobiology unit UGSF, and the breeding company Florimond-Després. So as we can see here, I was using in-gel bottom-up strategy since I was analyzing peptides. And the purpose of this project was to find new actors in the starch metabolism. So thanks to this analysis, I've been able to characterize proteins which were only known by their genome sequences. It was the first time we were able to characterize it as protein. And we also identified some proteins which were only discovered in Arabidopsis thaliana, which is the model plant for the studies on plant biology. So thanks to this analysis, we now have the most exhaustive proteome for the potato starch granule. So being able to characterize the protein composing a sample is one thing, but most of the time the interesting part is the quantity of each protein inside the sample. Fortunately, protein quantification can be performed by using mass spectrometry. There is several strategy for the quantification of the whole proteome. We can perform it label-free. We can use aquapeptides, which are peptides standards labeled with heavy isotopes. We can also use stable isotope labeling amino acid in situ and isobaric tags. So I will now describe these methods. So for the quantification label-free, the quantification is based on the signal intensity of each identified peptide for a protein. So with the intensity of each peptide, we can then have the quantity of each protein. The advantage of this technique is that it does not need further sample preparation for the quantification. However, this method is less accurate than label strategy, especially for absolute quantification, and it requires a pretty long computational time for data interpretation. Otherwise, we can use standard label peptides with heavy isotopes. So it is peptides which have C13 and N15 inserted in the peptide sequence in the amino acids. This will allow absolute quantification of the same native peptides which is inside the sample. So we have here the native peptides and we have the heavy isotope here. And so by comparing the signal intensity between the standard and the native peptides, we can quantify it. Another way to quantify is to use AV isotope to make AV isotope labeling in situ in order to label the proteins by feeding a cell culture with amino acid containing AV isotopes. AV stable isotopes such as C13 and N15. So samples are then injected in duplex in the mass spectrometer, and with the same principle as with the aquapeptides, we can compare the intensity between the light and the heavy peptides. And so this method is well suited for comparative proteomics when we want to compare one sample to another. Nevertheless, it is hard to apply on complex organisms which possess reserve organelles and organs. And finally, the isobaric tags is another way to compare several samples by injecting them in multiplex. This time, the quantification is not made during the MS scan. It is made during the MS-MS fragmentation because all the labeled peptides will have the same mass. But during the fragmentation, the mass reporter from the isobaric tag will be released. And so we will have here the peptide fragments, which will help to sequence the peptide, and the isobaric and the mass reporter will help to quantify it. So the intensity of the mass reporter will be used to quantify the peptides. So I made quantitative proteomics during my PhD in Lille in order to compare the concentration of each protein inside starch granule of different size. So the goal was to see if the quantity of each protein remains stable for each granule. As we can see on this diagram, at the contrary, the concentration fluctuates and even fluctuates a lot for some proteins based on the granule size. So let's now talk about the present. My postdoc project here in Bernal is about reusing the same protein A resin to purify multiple products. So shortly, protein A interact with the Fc domain of immunoglobulin G. And this characteristic is used in order to purify IgG in the industry, protein A resin constituting the first step in IgG purification. So when several products are purified, it is recommended to change the colon for each product. So the colon is discarded after a few cycles, whereas it can technically handle up to 250 cycles. So my job is to see if we can reuse the same resin for multiple products without significant loss in the product purity. To answer to this question, I will use mass spectrometry to control the presence of host cell proteins and cross-contaminations. In order to characterize the product itself, I will use a middle-down strategy since IgG are too large for top-down strategy. So to do this, I will separate the FAB region from the FC constant region using this enzyme, the SPEB. So this enzyme will chelate here between the FC and the FAB region, and I will use reducing agents in order to remove the cysteine bonds. To remove the cysteine bonds here, and so in the end, I will obtain the light chain fragments, the semi-FC fragments, and the FD fragments corresponding to the FAB region for the AV chains. And I will analyze it by MALDI, since MALDI is suitable for this kind of analysis. And finally, for the identification and quantification of the host cell proteins, I plan to use shotgun bottom-up strategy with LC-AZ cutoff and label-free quantification in order to obtain the quantity of each product, of the carryover, the HCP, and the protein A leaching. So we reached the end of my presentation. I would like to thank my academic collaborators, the industrial partners, and the funding bodies who make my postdoc project possible, and you for your attention.
Hi, Stanislas. Thank you for that. I'm looking if there's any questions in the chat or comments. While I'm doing that, Stanislas, could you? What's the cost of the column material in terms of industry? So they have to throw it out or not use it again after once. Is it after one cycle?
So for the costs, from what I remember, it's about I think it's $10,000 a liter for protein A resin. So yes, it's really expensive, and they discard it after. I will say it depends on the production indeed, but most of the time it's less than 10 cycles. So so yes, since the the colon can handle up to 250 cycles. So it's interesting for them to reuse the resin.
So if you can show that you can remove all of the protein, that there's no contamination really, you can help them to go to higher numbers of cycles. Is that correct?
Yes, the goal is to see if we don't have indeed any carryover when we reuse the resin. So if we can do it, if the industry can do it, can use the same colon to purify several products, it will reduce the price of the production.
Yeah. And do you have access then to a kind of a pilot scale plant in one of the companies that you could test your work, so that you could do it in a pilot scale rather than in a lab scale here?
For now, we are currently not at this step. We are still in a lab scale, but maybe in the future we will try a pilot scale.
Yeah. Okay. Okay. Thanks very much. I don't see questions in the chat. But can I just use this opportunity then to thank both speakers, and in particular, we're right on time. It's 1 minute to 1, so the schedule is very good. But can I just thank Stanislas and thank Sarah for both your presentations.